Jamie, take it away.
There we go. Can you hear me now?
Yeah, got you. We can see your slides well.
Okay, perfect. Thanks, Matt, for that introduction. I'm excited to tell you a little more about what Shrikhar previewed a few minutes ago in his lightning talk, and maybe give you a broader picture of the work going on in our project, which is titled Tools for Leveraging High-Resolution MS Detection of Stable Isotopes for Metabolomics Applications. So in addition to myself, the other co-PIs on this project are Doug Allen and Brad Evans at the Danforth Plant Science Center. And then the sort of boots on the ground are Javier in my lab at Vanderbilt. He's a PhD student. Shrikhar is a postdoc at the Danforth Center, who you just heard from. And then Jay is a programmer in my group at Vanderbilt, who's helping us with the tool development. And just want to thank NIH for supporting this NIH project. So before I kind of launch into the specifics of what we've been doing, I just want to remind everybody of, sort of a general aspect of metabolism, which is the function of metabolism is to convert nutrients into products and into energy. And so that is inherently a dynamic process, which involves transformation of carbon and nitrogen through metabolic networks. And one of the issues, the major issues we deal with in metabolomics is trying to infer this dynamic function from what in many cases ends up being steady state snapshots. And so just like any other dynamic process, we can look at a snapshot of that process, and we can try to make guesses about what's happening. Maybe here we might guess that the roads that have a lot of cars on them are very active, but other people might guess, well, maybe there's a lot of cars on the road because they're not going anywhere. In fact, if we had a way to convert those snapshots into a movie, we would actually see that where there's a lot of cars means that there's some kind of bottleneck and the cars aren't moving at all. So, there are ways using sort of temporal time series approaches like the previous lightning talk mentioned, where we can start to assemble snapshots into dynamics. But there are also other approaches like the ones that I'm going to be talking about today, where we can introduce a stable isotope, a nutrient that's labeled with carbon-13 or nitrogen-15, in order to help us trace the flow of atoms through networks. And so by measuring the incorporation of those stable isotopes into different downstream metabolites and measuring the rate of that isotope incorporation, we can actually make better informed guesses as to the activity of these networks and their fluxes. And that's really important for another reason, which is because metabolism, in addition to being very dynamic, is also very context specific. So even though many organisms have this exact same pathways and many of the same metabolites present, those pathways can operate in many different ways. And so, that's one of the most amazing things about metabolism is just how adaptable and flexible it can be. And one of the best examples of this is the TCA cycle, where, depending on the species or the stage of development of an organism or the availability of different nutrients, the TCA cycle can operate in completely different ways. In fact, it can operate in both cyclic or non-cyclic manners, depending on whether the central function is generating energy. For instance, a microbe that's growing primarily on sugar needs to oxidize that sugar to make ADP. On the other hand, organisms that receive a lot of input from amino acids into their TCA cycle may operate their TCA cycle in a non-cyclic manner and may export a lot of the carbon from the TCA cycle to make macromolecules, fatty acids and proteins and other things. And so again, depending on the species, depending on what nutrients are available, you may see completely different modes of operation. And so we would like to be able to use isotope tracers as a way to track the flow of those atoms and to be able to essentially quantify those rates or those different modes of operation. So the goal of our project has been to develop tools for analyzing metabolomics data sets that incorporate stable isotope tracing. And in particular, these are experiments where we provide isotopically labeled nutrients to living cells or living organisms so that the isotopic labeling is done in vivo. These can be carbon or nitrogen labeled metabolites. And they can be either singly or multiply labeled, depending on how we design the experiment. We would like to be able to collect samples from those experiments and perform mass spec analysis, either low-res or high-res mass spec data. And we'd like to be able to analyze those data sets to quantify, well, first of all, to determine which metabolites are being labeled, and then to quantify their isotopic enrichment. This is important because, out of all of the features we detect in a typical metabolomics experiment, maybe, only a small fraction of them are labeled by our tracer. And so if we're particularly interested in focusing on those labeled compounds, we can filter out a lot of the noise here and other unlabeled compounds right at the very beginning. Another important point is if we can leverage high resolution mass spec detection, we can deconvolute these mass spectra and separate our signal from interfering compounds. We can also, in the case of a multiple or a dual isotope labeling experiment, we can distinguish between, for example, carbon-13 isotopes versus nitrogen-15 isotopes supplied in the same experiment. And so our project has developed really two main categories of software tools as part of our research. On one hand, we've developed some targeted software tools that allows us to take a user-specified library of compounds and search the mass spec data for those compounds and accurately enumerate all of the possible isotopolog species that we expect and accurately quantify their isotopolog distributions, which are essentially the ratios of the different isotopologs for each individual compound in our target list. We also oftentimes need to be able to correct that data for natural background isotope abundance, and then finally to visualize the data. On the other hand, in other cases, maybe we want to do a discovery-based experiment, and we want to just search in an untargeted way through our data sets to look for all of the compounds or all of the features that show a signature of being isotopically labeled by our tracer. And so here we may just start with a list of features that we get from XCMS or some other pre-processing software, and use an untargeted workflow to detect all of those potentially labeled metabolites and to rank or categorize them based on their labeling dynamics. Then we can use other approaches to compare the molecular weights of those features that are detected, those potentially labeled compounds, to a network we would generate from KEGG that is both specific to our tracer as well as the organism of interest in our experiment, to try to help us annotate those metabolites that have been identified, that have been detected in our untargeted pipeline. And then finally, we're integrating all of these different algorithms and tools together into user-friendly tools like this mass spec aimbot tool that we're working on. So I want to describe the application of these tools to another example, in addition to the one that Shekhar mentioned earlier. Another example, which is the isotope-based metabolomics of soybean during seed development. And our main interest here is to try to maximize the value of the nutrients that are stored in the seeds during seed development. And here we're primarily interested in maximizing the protein and the oil content of the seeds. That's really what provides the value to the seeds and in their use as food or animal feeds, as well. If we were interested in some type of biofuel application, we would of course be interested in the oil. And so we're interested in questions like what determines the resource allocation during seed filling? How does the carbon and nitrogen supplied by the maternal plant get metabolized and redistributed into these main seed components: carbohydrates, lipids, and proteins? And one thing that's been very challenging in terms of optimizing this ratio is that if you look across many different varieties of soybean, the oil content of the seeds is sort of inversely proportional to the protein content. So we can sort of increase one, but there's a trade-off in that we decrease the other. And so one of the key significant challenges here is how do we break this correlation to actually find ways to increase both the protein and the oil content of the seeds simultaneously. And so for this test case, we've selected soybean lines that have altered protein and oil content in their seeds to allow us to compare those two varieties. So we have one that we call high protein plus high oil. So the total seed content is elevated in both protein and lipid. And then there's a high protein, low oil, which you can see here as well as over here, has less of both protein and oil once they reach the fully developed stage here at R8 compared to the high protein, high oil seed. And so we've performed isotope labeling studies during the seed filling period of development when the protein and oil are being made and stored in the soybean seeds. And we're interested in getting at this ultimate question, which is what are the challenges, what are the changes in central metabolism that account for those differences in oil and protein that we observe. So we'd like to be able to do these types of dual labeling experiments with both carbon-13 and nitrogen-15 tracers, and to be able to apply high-resolution mass spec to detect which pathways are active in metabolizing the carbon and the nitrogen from those specific nutrients. In this case, for example, glutamine that's supplied from the maternal plant. And as you can imagine, when we do these experiments, the complexity goes up even higher compared to a traditional metabolomics experiment, which is already a high dimensional data generating experiment. Now, for each compound that we measure, we're interested in tracking sometimes 10, 15 different isotopologues for each individual compound. So the dimensionality goes up tremendously. And if you talk about something like lipids, in the case of Shekhar's lightning talk, I mean, you can have 50 or more isotopologues for a given lipid species. So the dimensionality of these experiments goes up combinatorially as you increase the number of different compounds that you're trying to measure. And so because of that reason, and because of the general lack of software tools that are specialized for doing this type of isotopolog analysis, Most of the traditional isotope tracing workflows have relied on just more targeted MS analysis, where we have a list of where we have a biological hypothesis and we have a list of metabolites that we want to measure in a targeted way within those pathways of interest. And even with that hypothesis-driven narrowing down of the data set, there's still a tremendous amount of tedious manual processing of the isotopolog data that is required for analyzing these types of studies. And even though there have been in recent years some software tools developed that try to address these issues for processing isotopolog data, many of those tools still have shortcomings in that they can't handle multiple isotopic labels. They don't accept all the standard file formats. In many cases, they're command line driven tools that don't have user-friendly interfaces. They lack support in many cases for analyzing isotopolog data derived from high-res instruments or tandem instruments. And in many cases, they're only set up to compare a labeled sample versus an unlabeled sample, and they don't necessarily support time series analysis of the labeling data, which is incredibly important for getting at the dynamics of some of these pathways. So, we've sought to address many of these shortcomings in our project, and really one of the most overlooked issues in processing data from isotopic labeling experiments is just the difficulty in accurately quantifying the isotopolog ratios for a given compound. And that's because most metabolomics and mass spec software tools treat the different isotopolog traces as independent features when they do the quantification. And so what this means is that if you have some isotopologs that are more abundant, some that are less abundant, if the software is applying inconsistent parameters when it does the quantification, for example, it's using different retention time windows to integrate the area under these different peaks. Well, when you go to then try to quantify the ratios of those different isotopologs to each other, you can introduce errors that are as high as 5 or 10% in some cases, just because of the way the data is quantified. And so there's been some work done over the years to try to figure out how to do more consistent quantification of isotopolog data, which at the end of the day can improve the accuracy by more than 10-fold. One example of this is from Maciek Antoniewicz, who showed that just by making sure that you use the same fixed integration bounds to quantify all of the isotopologs in a given metabolite series, as well as making sure you use a consistent approach for baseline correction for each of those isotopologs, you can get substantial improvements in the accuracy of the isotopolog ratios that are ultimately calculated. Another more recent report came from Karsten Hiller's lab, where they described how doing peak shape correction by using the more abundant isotopologs within a series to help to correct the peak shape of the less abundant isotopologs could allow you to quantify these isotopolog ratios and quantify the isotope enrichments at much lower limits of detection, which actually becomes really important when you're dealing with experiments where the isotope enrichment itself might be very low. In vivo experiments with humans, for example, where the amount of isotope that can be administered is low for either cost reasons or safety reasons. And so being able to quantify very small levels of isotope enrichment can be really critical to these types of experiments. And so by combining these different ideas, we can ultimately generate much more accurate isotopolog ratios when we go to quantify the labeling data. And so just to kind of tie that back to our test case here, we had soybeans, data from soybeans that were labeled with either C13 glucose or dual labeled C13 N15 labeled glutamine. We had our two different soybean varieties, the high protein, high oil, or the high protein, low oil. And we did parallel labeling experiments with each of these different tracers, analyzed the samples with high res mass spec, and then processed it with XCMS to generate a list of features. We then applied our untargeted software tool, which we're calling Muisca, to automatically bin all of those features according to isotopolog series into compound groups. So the way this works is the software looks for features that have similar retention times and also appear to have mass shifts or progressions in mass that are consistent with incorporation of C13 or N15, as the case may be, and then groups those together into compounds and by binning those isotopologs and in some cases their adducts together. The next step is that the software then examines how those isotopolog ratios are changing over time. And in particular, it's looking for compounds whose monoisotopic mass is decreasing over time, and the higher weight isotopologs are increasing in abundance over time as a signature of compounds that are being labeled by our tracer. Then Javier developed a machine learning approach to classify compounds. He basically took all of the compounds in CAG and built used a support vector machine to build a classifier, which would take all of those labeled compounds that were detected by Muisca and filter them based on ones that appear to be real compounds versus noise. That classifier took into account both the molecular weight of the ion as well as the M1 to M0 ratio of the unlabeled compound to basically try to filter out those noise compounds from the hits we got. And then the last step in this untargeted workflow is to try to come up with a way to annotate those compounds, or at least to come up with suggestions for what the identity of those compounds might be that were identified, that were detected. So here, Javier's algorithm reconstructs a network from CAG that's specific to the organism in our experiment, as well as the tracer that's used in our experiment, builds all of those metabolites and reactions together into a network, finds those pathways that could potentially hold the compounds of interest that are being labeled by our tracer, and then tries to match up based on the molecular weight. What are some possibilities for what those labeled compounds could be, and then ranks them according to their proximity to where that tracer enters into the network. So for example, if we see a lot of labeling in this compound here that has molecular weight 116, it's more likely to be fumarate because fumarate is closer to where the glutamine tracer enters than some of these other possibilities. And so just as an example of how this can reduce the number of features that we have to sift through in our experiment, here's some of the results of applying this untargeted approach to the C13 glucose labeling data from the soybean experiment. So we pre-processed the data with XCMS, which produced about 25,000 total features, and those were binned. So we selected, so Muisca selected about a quarter of those features that could be bin together into an isotopolog series associated with a particular putative compound. And out of those total 6,000 features, they were bin together into about 1,500 total compound groups, out of which about a third of them were detected as being isotopically labeled. And then finally, out of those 500 labeled compounds, we could suggest IDs for about 200 of them by doing the comparison to CAG networks. So we've gone from a huge number of features to a smaller number of compounds of potential interest by filtering the data based on their isotope enrichment. And so here's just some example. We find, based on the C13 enrichments, using this untargeted approach, we find some pathways, for example, glycolysis, which has metabolites in it labeled from glucose, and the TCA cycle, which is labeled from glutamine. So we find some expected pathway activities that are labeled by these tracers, but we also find some unexpected activities. For example, we saw a lot of enrichment into the flavonoid pathway, which was probably something we would not have been looking for going into this experiment. And so by using this sort of unbiased approach, we're able to detect those different pathway activities. We may not detect labeling in every metabolite in those pathways, but sort of like mummy chog, we can use this untargeted approach to kind of focus us in on different pathways that are being enriched by our tracers. The nitrogen labeling data is also interesting for tracking the fate, for example, in this case, of the nitrogen derived from glutamine. So we detect enrichment of N15 in an untargeted way in many amino acids as well as nucleotides that derive their nitrogen from glutamine. And so once we've done that untargeted analysis, then we can take our data sets and analyze them in a targeted way. We developed a tool called Pyramid that can take raw data files and analyze them using some of the consistent quantification approaches that I talked about to be able to accurately quantify isotope enrichment using a target list that we provide the software. Also, we have another untargeted tool called Simple that Shakar has already introduced, which allows us to take data that's pre-processed by XCMS and do multi-label tracking of those isotopologs, compare labeling patterns across different experimental conditions, and do other types of clustering, visualization, and statistical analysis of the data. And so just to kind of show you one of the conclusions that came out of this study, we found using the labeling data from glutamine that we were seeing a lot of enrichment, a lot more enrichment in the organic acid species, especially those derived from the TCA cycle, but not nearly as much enrichment in intermediates like phosphoenolpyruvate that are upstream of the TCA cycle, even though we still see quite a bit of enrichment in pyruvate in the high protein, high oil variety. And so the conclusion here is that malic enzyme activity is in part responsible for this enrichment of pyruvate that we see in the glutamine labeling data. And maybe a better picture of this is shown here, where the data are pointing to significant enrichment of malate from glutamine carbon. So about 40% of the malate is derived from glutamine. In addition to supplying significant amount of carbon from the maternal plant through glutamine, a lot of the nitrogen, as we've seen for amino acid synthesis and protein, comes from glutamine. And so ultimately, because we see so much enrichment going into malate and fatty acids that's derived from pyruvate. But we see very little enrichment in PEP. We can distinguish the activity of malic enzyme from PEP-CK, which produces PEP directly, versus malic enzyme, which produces pyruvate directly. So this sort of supports an important role for both glutamine metabolism and malic enzyme in the synthesis of oil and protein. And so just to wrap up, we've generated a suite of tools for that are optimized for metabolomic studies that use stable isotopes. These are GUI-driven, user-friendly tools that can support both MS2 and high-resolution data. They can do, some tools are meant for untargeted analysis and others for targeted analysis. Some are for more basic users and others have more advanced features. But altogether, these tools allow us to generate and prove hypotheses regarding the in vivo metabolism of our experimental systems. We are also now really in a phase of trying to disseminate these tools. Simple as an R package that is available on GitHub and is also being integrated into the Metabolomics Workbench, as Shakar mentioned. Pyramid and Muisca are standalone MATLAB programs, which we are also going to disseminate through Vanderbilt's Tech Transfer Office and through links from Metabolomics Workbench. But all of these algorithms are being integrated into a single user-friendly tool called Mass Spec Aimbot that's going to be available as both a desktop and a web app version. So I'll stop there since I've used up all my time and just end on this overview slide here that sort of provides a big picture of all the work we've been doing in the project. Thank you for your attention.